Imagine there being two of you. Better yet, imagine there being two of me. While this seems crazy, chances are it is actually quite possible. Cloning, or the exact replication of an organism, is becoming more and more advanced. With this being said, the latest Gallup poll in May of 2017 found that 83% of Americans think human cloning is morally wrong. As this is an informative speech, I will leave the ethical dilemma of cloning up to you. One thing is for certain though, cloning is possible. I, Parker Eller, will be informing you of this scientific phenomenon. There are two cloning processes, artificial embryo twinning and nuclear transfer. And continued research on these cloning methods have led to unforeseen scientific advancements. To start, artificial embryo twinning was the first form of cloning. In today's scientific world, this is a very simple process. It follows virtually the same method that nature uses to produce identical twins. The only difference is that in artificial embryo twinning, the embryo is forced to split into two rather than naturally. If this split happens early enough in the developmental process, two identical individuals or clones will form. According to the University of Utah's genetics website, the first artificial embryo twinning occurred in 1885. Hans Dreich, a German biologist, used a remarkably simple experiment to split one sea urchin into two. When a sea urchin embryo is in the one-cell state, it undergoes a holoblastic cleavage, or egg division. After this egg division, the embryo is in a two-cell state with two genetically identical cells. When in this state, Dreich simply shook the embryo until the cell split into two genetically identical embryos. These two embryos eventually developed into two genetically identical sea urchins. This was the first ever cloning. Perhaps an even more commonly known experiment was Hans Spemann's research with salamanders. From 1902 to 1928, Spemann did a series of artificial embryo twinning experiments with the salamanders. He separated the two-cell embryo with baby hair. He simply crossed the baby hair between the two cells and tightened until the two cells eventually separated. Again, two unicellular embryos formed. These two unicellular embryos eventually developed into two genetically identical adult salamanders. This was a huge discovery as it was the first cloning of a vertebrate animal. As you have probably noticed though, artificial embryo twinning is quite a simple process. Subsequently, it only works for simple organisms. A more complicated process was needed to clone complex organisms. In 1952, Robert Briggs and Thomas King discovered the second form of cloning, nuclear transfer, using frogs. The textbook Life by Sadava explains that in this more complex process, the nucleus of an unfertilized egg is removed and replaced with the nucleus that contains the DNA to be cloned. In other words, an egg cell acts as a surrogate for the clone. The textbook Life also explains that this process is especially complicated because most nuclei in mature organisms can no longer activate the genes necessary for the developmental process. With this being said, in the experiment done by Briggs and King, an intestinal cell of a tadpole was used to create genetically identical tadpoles. This was a monumental discovery for the scientific community. A cell from an adult organism, rather than from an embryo, was used to create a clone. You may also hear this process referred to as somatic cell nuclear transfer. Somatic is simply the scientific word for body. This just means that a cell was taken from a living, mature organism to create a clone, which is exactly the process I just described. Nuclear transfer was used to clone several other organisms through the late 1900s. But the first mammal to be cloned was in 1996. Ian Wilmot and Keith Campbell, in one of the most famous scientific experiments of all time, cloned a sheep named Dolly. In the experiment, cells from the mammary glands of an adult sheep were transplanted into an enucleated egg cells. That just means the nucleus from the egg cell was removed. This egg cell was then transplanted into a surrogate mother, and eventually a baby sheep, Dolly, was formed. As I previously mentioned, it is hard to use body cells because most can no longer activate genes necessary for the developmental process. Because of this, the cloning success rate of the sheep Dolly was a measly 0.4% according to the University of Utah's genetics website. It took 277 attempts to successfully clone Dolly. However, the successful attempt resulted in a lamb that was genetically identical to the nuclear donor. The Dolly experiment led to the cloning of primates, humans' closest relatives, just 1 year later in 1997. Over the years, cloning has led to the development of unforeseen scientific and medical advancements. In 2013, somatic cell nuclear transfer led to the creation of the first human embryonic stem cells. These stem cells are pluripotent. In other words, they have the ability to become many different types of tissues. We can now use stem cells to help replace tissues that have deteriorated due to illness. According to the National Institute of Health, or NIH, stem cells can be used to treat blood and bone marrow disease, blood cancers, and immune disorders. As I mentioned, stem cells are created using the nuclear transfer process. Then these cells are allowed to develop until an early stage in development known as the blastocyst stage. In this stage, there are 16 cells. When in this stage, the cells are allowed to harvest on a culture dish. After the cells proliferate, they can become virtually any cell type in the human body. As you can imagine, by improving our understanding and development of stem cells, we can save many lives. As former US Congressman Jim Ramstad stated, embryonic stem cell research will prolong life, improve life, and give hope for life to millions of people. In conclusion, cloning has been made possible by the artificial embryo twinning and nuclear transfer processes. Further research into these cloning processes led to medical advancements such as stem cells. Scientists went from using the basic concept of twinning to replicate simple organisms like sea urchins, to implanting genetic material into enucleated eggs in order to replicate mammals and primates. By using these same methods, scientists and doctors can now use stem cells to cure many diseases. Also, by using these methods, scientists are fully capable of cloning human beings. However, due to ethical reasons, lack of funding, and lack of public support, a human being has never been cloned. What do you think? Should we start cloning humans?